This, as you can see here, let's see if we can turn it so it's going to work as well, is an extracted human brain from an individual who is willing to donate their body, as Sam was alluding to, and which includes their brain to the study of both science and education. And one of the things that we do in terms of harvesting this brain is we take it out as an educational tool for the medical school students as well as graduate students. Just to orient people before we jump into some kind of the nitty-gritty of neuroscience, this is looking at the brain from the top, coming down. This is the front part of the brain, which would be in and around where your forehead is. Then this would be obviously going to be the back part of your brain. Flipping it around here, you can see that way. And then these are obviously going to be the sides. Awesome. And one of the things you might notice in some of these specimens is there's a line that's kind of makes its way from one side of the brain to the other side of the brain. That is not typical, and it's not normal in a living human brain. This line comes about when we extract the brain from the cadaver, from the donor. As we cut through the skull in order to get that brain out, we have a saw, and you can imagine our skull is very thick and very strong, which it needs to be. For anybody that's had a fall on their head, you appreciate having a thick skull. And in order to get through that skull, we have to take a saw and cut through that thick bone. And it's not uncommon for us to cut through the skull and nick the brain. So that's what that line is there. So just to give you a little bit of orientation. So kind of starting from the very basics, our brain is divided up into a number of different lobes. And it's symmetrical. So there's a left and a right half. And you can see here. It's divided by this long, what we call a fissure here, which is essentially a space that exists to separate different sections of the brain. The lobes include the frontal lobe, as I mentioned at the front here. Then there's an intervening lobe in the middle called the parietal lobe. And then in the back part of the brain is what we call the occipital lobe. And then finally, the lobe that's on the side is our temporal lobe. That's next to the temples of the side of our heads. Okay? And the other main characteristic you can see that is one of the most important characteristics of the human brain is what we call the gyri and the sulci. The gyri are these characteristics up here, these convolutions that exist. In the human brain has the largest amount of convolutions of gyri relative to any other animal with one respect, and that's the dolphin. And the sulci essentially are the spaces that exist in between these gyri. And the reason we have gyri and sulci, as they are part of what we refer to as the cerebral cortex. Cortex means bark in Latin, so it's the outer part of our brain. And this is where the vast majority of our higher cognitive, higher thinking, higher functions occur. So things like memory, things like attention, things like sensory processing, motor processing. All that information essentially is generated and analyzed and processed in our cerebral cortex. So you can imagine the more cerebral cortex you have, the more functional capacity and behavioral capacities one is able to manifest. And the reason we have these folds, these sulci that exist down, and if I could actually take away some of what we call the pia mater that exists down here, you could actually just see how the depths of these sulci. But if you look in between here, I can pull this longitudinal fissure up here. The reason we have the main reason we have so many folds and so many convolutions is it allows us to have a greater amount of cortex in what is essentially a limited capacity size of our cranial vault. Okay? I think with that, I'll probably hand it off to Sam. I did forget about that. Yeah. Okay. So, so we talked about lobes as a kind of a fundamental organizational principle. One of the main tenets of neuroscience, particularly when we think about higher function, is the concept that was typically referred to as functional neuroanatomy. And functional neuroanatomy is at its core is the idea that function is housed in the cortex itself. And the idea being that the smaller the parcel of function that you can localize means the greater amount of functions that can be contained within the cortex. What do we mean by that? Well, I think the clearest example is when we look at, for example, the back part of the brain, as I mentioned, the occipital lobe back here. This area is what we refer to as visual cortex. which is a little bit ironic because it's the farthest part of the brain away from the eye. But the idea here is this area of the cortex that exists all around in this space here is where visual function is housed. So information comes from our environment through our eyes. It gets transduced or it gets converted from light energy into neural energy, electrical signals, which is something we may touch upon later on. And then that information gets conveyed through connections, through white matter connections, which is essentially can be defined as axons going from one area of the brain to the other area of the brain. And then it gets conveyed back to this area in the occipital lobe. And that information is then processed such that it makes sense of that visual information, that light information coming in from our environment, and it renders visual images such that our brain can understand. The idea here is, in terms of understanding this concept of functional neuroanatomy, is if I were to take some kind of electrode and introduce some kind of electrical current into that visual cortex, you would evoke an artificial percept of vision, right? Another example of that might be a little more clearer is if we look at going back into our frontal and our parietal lobes, is we have a sulcus here called the central sulcus, which you can see starts along the midline and courses down laterally across to the outer parts of the brain. Looking at this central sulcus, if you go towards the frontal lobe up here, this is what we refer to as primary motor cortex. And looking in the back behind the central sulcus here is primary sensory cortex. And what I mean by primary sensory cortex, it's the primary somatosensory area of our brain, such that if I were to touch my hand with a probe or my finger and so forth, I'm going to generate electrical energy, which gets transmitted up my arm into my spinal cord, and then gets sent up to my body. And essentially, it's not until those electrical impulses reach this sensory cortex that our brain can make sense of the fact that information is being touched, or we're having something touch our hand, okay? So it has to reach, essentially, it has to reach this sensory cortex in order to make sense of it, okay? And part of the research that I perform, or that is part of my research lab, is using, as I mentioned, neural technologies. And the main population of people that we work with is what we refer as our epilepsy individuals. And these are individuals that have electrodes that have been implanted directly onto their brain. And you may be wondering, why would an epileptic individual have electrodes implanted directly on their brain? And the main reason is because they are a surgical candidate. And the reason they would want to have surgery is because they are having medically refractory epilepsy, which means that their epilepsy is not being controlled by any kind of medications, or it's stopped responding to medications. In that case, if they are a surgical candidate, and there's a number of criteria they have to meet in order to become a surgical candidate, one option for treatment is a surgeon can go in and remove or resect out that part of the brain that's causing the seizures. And the goal is for them to become seizure-free. They can live a much normal life without having the worry of having a seizure. Well, the problem is that all epilepsy individuals typically have epilepsy arising from a different area of their brain. Okay? So what they do is they come in to the hospital and they get electrodes implanted on their brain, and then they get, in what I'm about to show you is the type of electrode which lies down flat on top of the brain itself. And then they have essentially their skull cap, which was cut out to be able to put these electrodes down. They have a skull cap get put back on, and they get wheeled out, and they hang out in the hospital for about a week. And while they're hanging out, the clinicians are recording the electrical signals directly from their brain. And the reason they would do that is because they're waiting for them to have a seizure. And as soon as they have a seizure, they can then, because they're recording those activities, they can then pinpoint with a fairly good accuracy and precision where those seizures are coming from. And then they can go in and they can resect out that part of their brain. You wouldn't want to resect out the part of the brain. You wouldn't want to resect any part of the brain unless you knew what you were cutting out, and that it would be of benefit to the patient itself. And so what I'm showing you here is an array of electrodes. These are called electrocortographic electrodes. And typically what they do is they will lay them down on top of the cortical surface like this. And then, as I mentioned, these wires would come out through the patient's skull, and they would get plugged into some amplifiers. From a research standpoint, while the patient's just hanging out, waiting and waiting to have seizures in the hospital, we can come in and we can plug our amplifiers into these electrodes, and we can record the electrical signals while they're engaged in doing various tasks. And one of the things that one of the research studies that we did a long time ago was to try to understand better this process of somatosensory processing in this, let me see if I can find it again, this precentral gyrus here, which is this sensory area. So again, if I touch my hand, it sends an impulse up through my spinal cord up to this part of the brain. And one of the things that allows us to do, as I mentioned, is we can stimulate this part of the brain. And when we stimulate this part of the brain, the person will have an artificial perception like they're being stimulated, like their hand is being touched, for example. It's actually quite interesting, I think, from a geeky neuroscience perspective. But when we stimulate the part of the brain that represents the hand that's on the cortex, people report a phantom sensation on their hand, as if they're feeling something, even though we're not making any kind of actual physical stimulus. But the sensation that the people report is completely variable. People report like a sense of like wind brushing across their hand, or they report, this is when we're stimulating this part of the cortex, or they report it feels like sandpaper rushing up across their hand. Sometimes they even report a painful stimulus, like it hurts, like a needle's being impressed in their hand. We're not touching their hand at all. All we're simply doing is just stimulating part of their brain. So we ran an experiment to test out the idea of what would result in a faster reaction time. Stimulating their hand where they're reporting a sensation coming from, or stimulating their brain directly. If the question is press a button every time you feel a sensation, what would the hypothesis be for what would be result in a faster reaction time? Well, our initial hypothesis was when you'd stimulate the brain, it would result in a much faster reaction time because you're bypassing that ascending axons that go from the hand up the arm up to the spinal cord up into the brain, which takes time. It takes about 35 milliseconds for that impulse to get up there. That indeed is not what we found. We actually found that when you stimulate the brain itself, that it took much longer, on the order of about 100 milliseconds, for the person to press a button to indicate that they felt something with respect to touching the hand in the same spot where they felt that phantom sensation. We are completely puzzled by this. And there's a whole bunch of hypotheses, and I won't get into the nitty-gritty, but the application here, and one of those things that we're really interested in is prosthetic limbs. So prosthetic hands, prosthetic limbs is a vast and rich market that is really having great benefit for people who have lost limbs, for example, in combat. And one of the things that is very, very important is if you can't feel something touching your prosthetic hand, you're never going to be able to have a very effective prosthetic hand. So how do we introduce stimulation of the prosthetic hand, and how does our brain interpret that information? And if there's a discrepancy between the time it takes for that prosthetic hand, the stimulation to get to the brain versus how long it takes for the brain to process that information, that's going to result in a pretty poor performing prosthetic hand. So these are the types of research questions that we ask, and this is the types of electrodes that we use to try to ask those types of questions.
We'll switch up the samples now. All right, so let's move from the outside of the brain into the inside of the brain. So unfortunately, in the 20 or so minutes we have left, we are not going to be able to cover everything, surprisingly perhaps. But what we can do is we can highlight some pretty cool examples. So you just heard a lot about how those larger lobes of the cortex are responsible for certain types of sensory processing, when you might be wondering, well, how do you actually get that information out of the brain, right, and out to your limbs to actually move your hands or your feet and do a little dance on a stage, whatever you need to do. Well, if you look on the inside of these samples, what you can see are these really large, what look like tracks, right? We see them right here. And you can also see really large tracks here coming down. These are the actual axons, or bundles of axons, that are leaving the neurons from the cortex. The ones that we're looking at here are called commissural fibers. These are fibers that actually only go between the hemispheres, so between the left hemisphere and the right hemisphere. And they allow those two hemispheres to communicate with each other. Conversely, over here, what we're looking at are these axons that are running out and down to the brainstem and the spinal cord. If you look at this, it's actually really quite, let's look at it this way, really quite beautiful. This area right here is called the corona radiata, which means the radiating crown, because it might look to you, right, maybe if we can get this up here a little better, like a crown on top of the brain. And this area right here is the starting point of those axons that are leaving the cell bodies that are up here in what we call the gray matter of the neocortex and becoming what is called the white matter. So gray matter, right, gray matter journals are the cell bodies right here that are responsible for all of the behavior that you've been hearing about. These axons are going down through the corona radiata, through something called the internal capsule, and all the way down to another part of the brain we call the brain stem, past what we call the medulla, and down to your spine. And we'll show you some spinal cords in a second. But these large bundles of axons are capturing all of that information and sending it on out. In the case of, let's see, what side are we on here? This is a left hemisphere. It would actually be controlling the motor movements of the right side of your body. So that's right, the left side of your brain controls the motor movements of the right side of your body. And it does this by transiting through a whole host of what we call descending motor tracts. Perhaps the biggest, and I'm not going to say the most interesting in case there's any other neuroscientists in the crowd, but a very interesting one is the corticospinal tract that comes from this neocortex, crosses the midline, and then controls the motor actions on the other side of your body. I think I will also show a little bit more detail here, this area. So we've talked a lot about the cortex, right, and the neocortex. What we haven't talked about is the cerebellum or the brainstem. And those are the three major ways that we sort of break up the central nervous system. We also consider the spinal cord in the central nervous system, but we'll get to that in a minute. All of, for example, the cranial nerves that are responsible for everything from the neck up in terms of sensing things or talking, moving your tongue, whatever, are found over here in the brainstem. And the brainstem itself is broken up into three different areas, and I'm going to show you in detail very quickly before moving on. One of those, called the midbrain. Okay, you have to be very good at three-dimensional space to follow what I'm about to say. Let's come over here. So right now, what you're looking at are slices of a brain, right? This one right here is a horizontal slice. So you can see that we're basically, if we were to cut the brain from the top down, we're looking at a horizontal slice. This one is what we call a coronal slice. If we were to slice the brain from the front, from the anterior to the posterior, it would look like this coming through. And what's very interesting about this particular set of slices is they intersect at the midbrain, which is the topmost part of the brainstem. And you can see it right here and right here. And if you look very closely, you can see, actually, I guess I'll make it easier for you to see. Here you go. You can see these slightly darker areas right here, and these slightly darker areas right here. Again, this is cutting from the top down. This is cutting from the front to the back. And this is the part of the midbrain that contains the dopamine neurons that you've probably heard a lot about in various contexts. The dopamine neurons are found here in an area called the substantia nigra, and they can actually be seen by eye just by the different coloration of the cell bodies of those dopamine neurons. These are the same neurons that are susceptible, for example, to Parkinson's disease, right? When you have neurodegeneration of dopamine neurons, that's what leads to the inability to control your motor movements. We have several treatments for it, and maybe actually Kurt would be better to talk about them than me. But this is an area that's received a great deal of study, because one of the questions that I think we've all had for a long time when it comes to Parkinson's is, why those neurons? Why are they more susceptible to neurodegeneration than others in the brain? And if we can figure out why they are, maybe we can find a therapeutic intervention. We don't have it yet, but we now have a really, really good understanding of the molecular landscape of these different types. There's actually many different types of dopamine neurons within the substantia nigra, and which of those types are the ones that are ultimately most susceptible to that cell death. And so this is all happening in the brainstem at the topmost part, which we call the midbrain. Below the midbrain, we can look at it right here, this is pretty cool, is the pons. And below the pons is the medulla, which we also typically break up into the more rostral, or I guess I would say the more dorsal part right here, which is the classical medulla. And then we call the bottom part, as it gets really, really close to the spinal cord, the caudal medulla. And lots of really interesting and crazy stuff goes on there, and we could spend a whole couple of weeks talking about it. And I see some people from my class here, and I know how excited you are to do this in a couple days again. Yeah. Okay, so with that, before I get too into it, Kurt, do you want to maybe, I think it's a good example maybe to show some of the DTI right on top of this.
So these are fascinating specimens, but how do we look at them in individual people without actually having to die and donate your brain? Well, we can use modern day imaging technology to characterize and reveal and then explore these types of large-scale types of white matter tracks, these connections in the brain. And the most commonly used approach for that is a technique that comes from magnetic resonance imaging, or MRI. Many of you may have had an MRI. It's an application of an MRI technique called diffusion tensor imaging. And what you'll see here is one of the things you can use DTI to do, let's see if I can turn it so it makes sense, is you can quantify and you can explore, as I mentioned, I didn't do anything at all. There we go.
You've got to be good at spatial orientation.
Yeah, evidently I'm not. So this is so what you can see here. This is an image. The gray in the background is a typical MRI anatomical scan that allows us to explore the basic anatomy of the brain. But then if you look at down here, for example, this is an area of white matter, as Sam was just alluding to, which is composed of bundles and bundles and thousands and hundreds of thousands, if not millions, of axons. Well, just looking at that white matter there in this image, you can't explore, you can't understand the types and directions of tracks that exist, the white matter axons tracks that exist. So through this technique called diffusion tensor imaging, it allows us to track the movement of water molecules in the human brain as they course down a bunch of bundles of axons. Because axons have a given direction, as you can see here in blue, and as Sam was talking about these cortical spinal tracks, water courses down, obstructs these fibers to represent where those axons and the directions of the axons exist. So this cortical spinal tract, as Sam showed you, it was the image, the specimen he had was a mid-sagittal cut, or approximate mid-sagittal cut, so it cut down the middle, and it was dissected out, so you could see these axons going that way. Well, this is a non-invasive way of showing off the exact same thing, and these same fibers going this way. Here are those corpus callosal fibers, the fibers that he was talking about, where they go from left to right. And then finally, there are fibers that go from they go from the front to the back. And you can see those are these kind of orange and green fibers, which is hard to appreciate on this coronal cut. So if you compare this image to the brainstem, or to the actual kind of cut, you can see this is a way that scientists, as well as clinicians now, have the ability to understand where axons are going from and to in any given individual's brain. And one of the volunteers that we have that's going to be talking on the panel later this evening, Dr. Andrew Coe, is a neurosurgeon. And he uses these types of images to understand where these axons are in an individual's brain in order to try to avoid cutting through them in order to try to prevent postoperative damage and postoperative loss of function.
I think in the time we have left, before we take questions, we can now continue on down past the brainstem, all the way down to the spinal cord. And here we have an example, and I'll show you another one in a second, of an intact human spine. So right off the bat, I just want to highlight how we have this very strong structural fiber all around it, right? And I think you can actually see it beautifully right here before we open it up and see the spinal cord. These are the meninges. They would also be around your brain proper, but we've removed them in these samples so you can see the cortex underneath. And this sample is still on it. These are the meninges that encapsulate all of your central nervous system and provide a protective layer. They're also responsible for containing your cerebrospinal fluid, which is constantly circulating throughout your central nervous system and moving around all of the different metabolites, and also making sure that all the stuff that shouldn't be in there ultimately gets filtered out and cleaned out as well. All right, so if we were to remove these meninges in the spinal cord, we can see some pretty cool stuff. In this sample, we're a little more caudal, more towards the tail. We can see something called the cauda equina, right? These are actually the spinal roots that are just past where your last spinal vertebrae would be. So right over here would be your sacral vertebral segments, so those are the lowest ones down your spine, and just past that are these nerve roots. These nerve roots are the area, if you've ever had a spinal tap, or know anyone who's had a spinal tap, for example, would be the area where your physician would be aiming their needle to collect a sample. And the reason they would do it there is, as you can see, in this region of all of these nerve roots, there's not a spinal cord. Whereas if you were to stick a needle, for example, directly into the spinal cord, you could probably imagine there'd be adverse events with that damage. So looking at this here, you get a really, really great example also of just how large these nerves can be. I mean, that's I think, pretty clear right there. The other thing that's really neat in these samples, and I'm going to put it pretty close to here so you can really see it, or maybe not if it doesn't zoom in. As you can see in this coronal view, or transverse view, sorry, of a horizontal view of the spinal cord, you can actually see all of the different parts. Unfortunately, I don't know how clear that is. You can sort of see how there's a whiter area and a darker area. And what that is delineating are where those white matter tracks, those axonal tracks, are going down the spinal cord or up the spinal cord compared to the neurons that are actually found in what we call the ventral or dorsal horn of the spinal cord. The ventral horn, which is the part, we'll start with the dorsal horn. The dorsal horn, which is the part of the spinal cord towards your back, contains the sensory neurons. So all of that ascending information that Kurt talked about, coming in from your hands or from your toes, comes in to the dorsal horn of your spinal cord. Some stuff happens, and then those motor commands come out of the ventral horn. And you can sort of see that if you look. I mean, I wish I had a better example of this, honestly. You can sort of see that in some of these slices. Is there anything else about the spine you wanted to?
Yeah, I just wanted to give everybody a relative scale here in terms of the size of the spinal cord that we're dealing with. So you can just see how big, how small it is.
Oh, yeah.
There's
Great.
a dime. So if you, I mean, it's small, and you may go, oh, that's pretty cool, but to appreciate that, all of our sensory and motor information, as Sam was just discussing, courses up and down this tiny, tiny little structure. Every muscle we move in our body, outside the face, every sensation that comes up from our skin, that goes to our brain, courses through this tiny little structure. And the perspective here, in relation to that dime, just tells you how small it is. So it tells you how catastrophic it is when somebody has a spinal cord injury, because you can easily damage a good sector of that spinal cord and result in catastrophic functional loss or functional impairment. And that's why, I guess we don't have any vertebrae here, but if you look at the vertebral column itself, it's housed in very thick, heavy protective bone. And it's one of these structures where it can be damaged easily, and even small amounts of damage can have widespread catastrophic injury.
So that's it. You're all neuroscientists now. Congratulations. We're going to be able
to sign your documentation.
Yeah, absolutely. If you have any questions, now is your opportunity to ask us. I think we have about 10 more minutes before we need to clean up and make way for the panelists here. Don't be shy. I'm sure we've said something that might have confused you, or maybe you've heard something that you'd like clarified. Again, that is not of a personal nature. And if so, please, speak up. Yes? The meninges part? Or you mean the things coming off to the side? These right here? Okay, great question. So we've talked about the central nervous system, right? There's a whole nother nervous system called the peripheral nervous system. What we're seeing here as we go down the spine are the ganglia, or collections of cell bodies of that peripheral nervous system. Each one of these, basically, each one of these roots that we see coming off houses one of those ganglia inside of it. Again, we're looking at the meninges now, so it's all covered up, right? It's all covered up by these meninges. But that would be the sensory information coming in from the periphery before it enters into the spinal cord and then ascends up to the central nervous system. Yes? Sorry, louder? Yes, right here. These are just exposed roots. Yeah, exactly. So there's 4 main parts to your spine. There's the cervical, the lumbar, the thoracic, the lumbar, and the sacral. And that ends right, at the base of your butt, essentially. Or a little higher, I guess I would say. A little higher than that, yeah. And right below that is where the cauda equina is, which are these spinal roots. And that's the area that a doctor would be putting that spinal tap or doing a spinal block inside of. Yeah. But those are outside of the vertebral column. I think that's technically correct.
The cauda equina?
Yeah.
They're actually inside.
Oh, inside. Ah, got me. Yeah.
Yep.
So with the spinal cord injury, how is it determined what part of
So clinicians, doctors have lots of functional tests they do. One of the things Sam was talking about is these different levels. So we look at these dorsal ganglion here, which is their cell bodies, and they come off, each one of these ganglia here come off at a different level. And as he was alluding to, we've got a set of cervical levels, thoracic levels. You may have all heard about the lumbar and the sacral levels. And it's just, where those nerves are coming off the spinal cord is just kind of serial up and down that spinal cord. And what they're doing in terms of a sensory aspect, so if I touch my arm here, I feel the sensation coming from here. And that corresponds to what's referred to as a dermatome. And a dermatome essentially is just a mapping of the space on your body that corresponds to where those nerves are going into the spinal cord itself. And the way they work it is they will test out which dermatomes patients can feel. So if you're working it, so this is our thoracic cavity, which is where our heart and lungs are, has 12 levels. And so they'll work their way down, and they'll do a little touch and say, do you feel that? Do you feel that? They'll just kind of go down, and they ask the patient, you're just asking the patient, do you feel that? That's essentially it. That's the basics of it. And you go down to the point where they can either no longer feel or they can start feeling those sensations. So if you have a high injury, you can imagine if you have, it's a complete injury, then all of a sudden you're not going to be able to feel anything coming up because that information is coming from your body is going up. It has to get to the brain for you to be able to feel it. But if you have a lower injury, then the things that are above that injury are still sending signals up to the brain. And the patient would be able to report that. So the idea is you go down to the point where they can no longer feel something, and that approximately is at the level of the spinal cord where they would have an injury. That's one example of a test that they have.
And just to add to that, in this one, which we haven't really looked at yet, if you look closely, all right, so this is going from closer to the head to farther from the head. There are two areas along here that are slightly thicker, right? We call that the cervical enlargement, which is up here, and we call it the lumbar enlargement, which is down here, and then it's a more constricted, thinner waist between the two. And those two areas of enlargement correspond with the segments of your spinal cord that are responsible for controlling your arms and your hands and your legs and your feet, right? Where you have a lot of sensitivity, right? You can read Braille with your fingertips. You can have high resolution. If you tried to read Braille with the side of your stomach, you would not be as successful. And that is because of these enlarged areas being able to house all of the machinery that's needed to make sure you have that resolution both in terms of sensory input and also in terms of motor control output. And so those are two of the more important areas along. I mean, it's all important, as Kurt said. But for dexterity, more important for dexterity.
Yep.
Great questions.
As someone who's experienced a cervical spinal injury before, I've always been curious about the tensile strength of the spinal cord, or is it really just our bones protecting it? Like, how strong is the spinal cord?
So you're referring to if there was like a shearing injury to the spinal cord itself.
Yeah, like, could you just rip it? You know?
Well, you could rip it. So if I'm understanding your question correctly, it's can you put strain on the spinal cord without having any kind of noticeable or any kind of injury or impact?
Yeah, like how much could the spinal cord handle if not for its protection with the bones in your back?
So I'm going to answer that with that is above my pay grade. But if Dr. Andrew Kodesh, when he shows up, save your question and ask him because he would be able to answer, I think, that type of question.
And as an addition to that, you're exactly right. The protection really comes from the lamina within the vertebrae, which are essentially cartilage that allows you to pad the impact of any potential, tomfoolery and tries to prevent that damage from occurring. Someone had their hand up right here in the front. Yeah.
I'm wondering, within the spinal cord, is it only the axons of the neurons, or does it consist of cell bodies as well? And then why?
It's a great question. It's both. So in the spinal cord, we have these large tracts of axons that are coursing up and down, but we also have large collections of either sensory motor or this other third population called interneurons that are all responsible for basically making the computations necessary to get that sensory input and that motor output. Now it requires, not always, often requires input from some cortical region as well, unless it's like a reflexive response. But yeah, it has both neurons, gray matter, as well as axons, white matter in the spinal cord.
I think one really interesting application to this concept of having local circuitry in your spinal cord is there's a concept, a well-established theory called the descending modulation of pain theory. You can imagine if you're walking through a forest and you hear a twig snap, and you look back behind you and there's a wolf that's staring down at you and it's with blood dripping from its fangs. You're like, I got to get out of here. So you start to take off and you step on, say, a twig that goes right up your foot. Well, that's a situation where you would have lots of pain and you would want to stop and try to pull that out. Well, you probably would pull it out, but the idea is you need to hightail it out of there, right? You need to get out of there because you don't want to get eaten. So this theory, this concept of the descending modulation of pain is there's an area in our brainstem, as Sam was talking about, which actually sends axons back down to the spinal cord. So when pain signals come up, they reach this part of the brainstem, and there's some local processing that occurs that makes you realize that I don't want to feel this pain right now. I want to survive. I gotta make sure I get out of here, so I don't want to feel this pain. So these axons get sent back down to where those pain signals are coming from, and actually sends little neurotransmitters to block that pain from coming up the spinal cord and to reach your brain, so that you can hightail it out of there and not worry about that pain at that point in time. And once you get to a safe area, then all of a sudden you can feel the pain as much as you want.
I think we can all relate to being chased by a wolf.
Yes, with
in that
blood
particular
dripping
scenario.
from its fangs.
Yeah. Any other in the middle? Could you, so is the question, could you have autonomic? Well, so if you were to disrupt, for example, the motor neurons that are controlling the diaphragm, you would not be able to breathe. So there could certainly be some edge cases where some weird stuff happens that I'm not familiar with. But typically at the level of the injury on the spinal cord, we'll have an impact of the physiology at that level, at those dermatomes and myotomes that you were already hearing about. Yeah. In the middle.
What's kind of the scale of the area that a single dorsal root ganglia takes sensory input from?
Well, so it depends on the dermatome itself. But so if we take, for example, the T4 dermatome, which is around where our nipple line is, it goes all the way around. So the T4 dermatome, it gets a little complicated in terms of the circuitry, but it's about this wide and wraps all the way around from the front to the back. So that one side, so my left side is going to be sending impulses into the left T4 dermatome or left T4 level in the spinal cord, and the right side is going to be sending it into the right side of the spinal cord. But if you look at your hands, for example, as Sam was alluding to, the dermatomes are much smaller there, for example. So it just depends on, and it has to do with how many sensory receptors are in that patch of skin. It has to do with a couple other kind of big factors, but they do code for the information that would, for example, that would come from this area all the way around to the back. It goes back to that midline.
And so a good example of that is at the back of your thigh is an area where you have a large area with very low sensitivity, right? You could have two tweezers poking you, and you would only feel one poke, whereas that would not happen in one of these other dermatomes that has more representation, for example.
I saw a hand over here that somebody, yep. That is a very amazing question. So just to reiterate for those that couldn't hear, when looking at these wiring diagrams of the human white matter pathways, how does one determine which ones to sever, which ones to cut in terms of a clinical application like the removal of a tumor? Well, there's no easy answer. It depends on where the tumor is, it depends on the size of the tumor, it depends on the prognosis of the resection of the tumor, and it depends on what functions that would be impacted or what functions could be potentially lost by cutting through those axons themselves. So oftentimes surgeons refer to what they have a concept they refer to as eloquent cortex, which generally refers to speech and motor function. And those are the types of functions that you really want to try to avoid impacting. So many of you may know that language for the vast majority of us is a lateralized function, so it's lateralized to the left hemisphere. And if you cut through the pathways that connect various regions of the language pathways, if you cut those connections, it's not surprising your patient's going to have potentially language impairment. So you try to avoid cutting through those. But if you have a tumor that's growing, and if you don't remove it, that they may die, then you have to weigh the priority of, let's get that tumor out of there and worry about the postoperative impairment later. But if the tumor is not growing and it's benign, then maybe you can just cut out less of it and preserve those pathways, for example.
So these were fantastic questions. Thank you. We hope this was informative and interesting, and maybe more than anything spoke a little bit of your curiosity. And if you have any other questions, feel free to reach out to Gray Matters, and maybe they can direct you to one of us to talk about it. But with that, thank you for your time, and we're now going to clean up and set up for the panelists.
Thank you so much, Dr. Sam Golden and Dr. Kurt Weaver. Let's give them another really big round of applause. We will now have a brief intermission before we get ready for our long-awaited panel. So use the restroom, grab some water, and come right back. Just wait for them to get up. You ready? If everyone could start taking their seats again, that would be great. All right, welcome back, everyone. Thank you so much for being here. And welcome to an evening with Neuroscience. We hope you've enjoyed the pre-panel events upstairs and downstairs and the mini symposium, the brain demonstration so far. And now we're looking forward to a very lively panel discussion. My name is Emma.
I'm Sarah. I'm Isabel, and we'll be your hosts tonight. We are beyond grateful for your presence, and also to all of the members of UW Gray Matters who have been planning this event for months.
So, neuroscience is a field that is often pretty daunting and inaccessible to the public due to complexity and fear that surrounds difficult scientific language use and concepts. If that wasn't enough, socioeconomic factors, including the high costs of accessing scientific papers and just a lack of diverse perspectives within the field, just make those barriers worse. So Gray Matters Journal, which is our nonprofit founded here at the University of Washington, works to amend these problems by bridging the gap between neuroscience and the public, uplifting underrepresented voices and inspiring the next generation of scientists and professionals in the process. To do this, we produce a high-quality journal issue each quarter that is written, edited, illustrated, and designed entirely by undergraduate students. In addition, we hold large events such as this one, and we lead outreach initiatives to reach even more people in the public. Our organization continues to grow with every edition, striving to serve the mission of bringing neuroscience to the public in an accessible and accurate way.
Over the last year, Gray Matter has hosted numerous outreach events, bringing both entertaining and accessible neuroscience education to over 3,000 students at local Seattle schools. These operations are made possible through the volunteer time of hundreds of undergraduate students, and such we deeply solely on the community support to make these events possible. As you enjoyed tonight's evening with Neuroscience, please consider supporting us with any donations during the intermission or at the end of this event. We greatly appreciate any and all contributions that you can make.
An evening with Neuroscience is always a special event, but this year we want to take a moment to acknowledge the position we are in today, hosting a large science-centered event in a challenging science environment. Tonight, we have an opportunity to learn and to celebrate both neuroscience and scientific work. We hope to deliver an exceptional learning experience and also dare to recognize the crucial, beautiful, awe-inspiring role that science plays in our lives. So far this evening, we have showcased research, art, and had a close-up view of the brain and spinal cord. Now, as we move to the panel, we will examine neuroscience from multiple perspectives and reflect on where we've been and where we will go. We have 6 incredible panelists here to discuss neuroscience and to answer audience questions. From your left to right, we are honored to formally introduce you to Dr. Nino Ramirez, Dr. Staci Sorensen, Dr. Jessie Miles, Dr. Michelle Chee, Dr. Kyobi Skutt-Kakaria, and Dr. Andrew Ko. We will begin with around 50 minutes of prepared discussion, and after a brief intermission, we will return for an audience Q&A where the panelists will answer all of your questions live. And without further ado, over to our panel. They will begin with a round of introductions.
Okay, perfect. Thank you very much. It's so amazing that you organized this. And you can't imagine how happy I am to see so many people here. When I drove here, beautiful weather outside, Friday evening, people were partying everywhere. I thought, who comes in here into this dark room to talk about neuroscience? But I'm totally with you. I think there's never been a better time for neuroscience. And every morning I get up and think, wow, I would have liked to be now a graduate student. Because, I mean, you have tools, you have opportunities that are just amazing. So I'm really looking forward to this session today. And so my name is Nino Ramirez, and I'm the director for the Center for Integrative Brain Research at Seattle Children's. And we do all sorts of work on epilepsy, but also the neural control of breathing and the control of the heart, actually. So that's me. Staci, why don't you start then?
Okay, how's that? I'm Staci Sorensen. I work at the Allen Institute for Brain Science. I am the Associate Director of Neuroanatomy, and we spend a lot of time trying to figure out all the cell types and circuits that make up the mouse and primate and human brain. And I'm excited to be here today. I spent a lot of time at UW as an undergraduate and then as a graduate student, so it's pretty exciting to be back on the other side of the room. Yeah, looking forward to your questions. I'm really excited to see so many people here. Thanks for coming.
Hi, everybody. My name is Jessie Miles. I'm a postdoc at Seattle Children's Research Institute and in Grid Lab here at UW. I study human brain development, and in particular, I study activity changes throughout the brain as kids are aging at different developmental stages. But I think probably the main reason I'm here today is because I'm one of the founders of Gray Matters, and have finally gotten to the stage where I've done enough to be here with everybody else. And so yeah, it's great to be here, and like Staci said, being on the other side of things is quite a privilege and honor. So thanks for having me, and I'm excited to chat with you all and answer your questions.
Hello, I'm Michelle Chee. I'm glad to hear I'm not the only one surprised to find themselves on the other side of things. I think I'm one of just 2 clinicians on this panel. I'm a general neurologist at the University of Washington. I graduated residency just last year, so I've been attending for like 10 months. Thank you. And I think other things that I'm interested in and that I think we'll get around to talking about are health equity. I've done some work with Partners in Health recently, through which I've met Chloe Darlene, who I think some of you know, and in particular, equity for patients who don't speak English as their first language.
Got it. Yeah. Hi, I'm Kyobi. I teach for the neuroscience major. I probably have had like a third of you in my classes. I would say there's nobody more surprised to be on this panel than me 20 years ago. It's been a long, circuitous path that led me here, and my trajectory through research has led me back to wanting to also study human brain development, but of all of you and developing all of you into young neuroscientists. So thank you all for being wonderful students, and I'm excited to be part of the panel. Thank you.
Hi, I'm Andrew Coe. I'm a neurosurgeon here at the University of Washington. I'm a functional neurosurgeon, so my specialties are epilepsy, movement disorders, probably the most neuroscience-y part of neurosurgery. Although I have to admit being a little bit surprised to be up here because compared to real neuroscience scientists, it's a little bit of a different path to get here. I was actually a philosophy major as an undergrad, so wound up here again a little circuitously, but yeah, just I think it's great when you get people interested in how the brain works and how you can. I mean, from my side, it's more like what can you do to it to make people feel better and or treat some of the problems that we run into. So getting started, you need the scientific basis for a lot of the stuff we do, and it's always great to work with people who do the nuts and bolts. But yeah, thanks for having me.
Perfect. Okay, so we go now to a few questions that were actually prepared by our amazing organizing team. And I told you that there's never been a better time for neuroscience. And the reason is there have been so many innovations. So the first questions was to the panel was, how are innovations in your field assisting your research? And also, what's your favorite innovation that you see? So who wants to start with that, answering that question? Okay, Kyobi.
Well, I want to jump in front of Staci because Staci is better at this than me. So the innovation that's really been exciting to me over the last 10 years or so is connectomics and mapping out the connections between neurons in the brain. So my field was Drosophila neuroscience, so fruit fly neuroscience. And at this point, we now have connection diagrams where you can count the number of synapses between every cell in the brain. Within a year or two, we will now have the entire brain, both the forebrain and the ventral nerve cord, the spinal cord of the fly. And I think that is driving modeling and driving questions in a way that is just incredible. The speed of progress in Drosophila is really mind-blowing to me. So I thank you for the work that you all are doing.
Kyobi, you're so right. I was thinking Staci has to start this because I tell you, I was an invertebrate neuroscientist because when I was young, you couldn't identify single nerve cells in a human or mouse brain or in a mammalian brain, but you could do it in invertebrate. But now through RNA sequencing and all these things, we can do things that are actually at the identified levels. And I think the masters are sitting here. So Staci, why don't you talk about your innovations a little bit?
Okay, well, that's just a little bit intimidating to live up to all that hype. Thanks, guys. My son would say you're glazing me. But yeah, so hopefully people saw recently that we just released this cubic millimeter volume of the mouse brain where we, mapped that the synapses of 100,000 neurons or more and can characterize the organization of the brain at a level that we have never been able to do before. And actually, it's just the first step. We're now in the process of going from a cubic millimeter to a whole mouse brain or a large chunk of a primate brain. And yeah, there's no stopping. I think one of the things that I personally contributed to that, the release of that data, was to integrate the cell type definitions that we get from the anatomy with other ways that we have studied the brain over time, gene expression, their electrophysiology, their excitability, their shapes, and then using those properties, we've learned a lot of other stuff about neurons, when they're active, how they function, in different situations, and then by using the shape of neurons, we can, we have been able to take a lot of those other modalities and then map them onto this detailed map of the anatomy of the brain. And so I think that trying to integrate data sets and integrate modalities that give us the possibility of taking the gene expression profiles of the neurons to connect from the mouse cells to the human cells has been really fruitful, so that we can take all this stuff that's really cool and looks amazing and then translate it into something that can actually advance human health and well-being. And so so yeah, hopefully you'll look out for it.
Yeah, so let me just explain to you, like the connectomics basically looks at the synaptic network, the hardware where how neurons talk to each other. But I told you that I would love to be young right now, because really this opens up a lot of new questions, and we haven't really addressed them. And I just come back from the University of Utah, where I heard a talk from Corey Barkman. She works on C. elegans, and C. elegans has like only 312 neurons. I might be wrong with this 300, but around that amount, okay? And she talked about how these neurons communicate via peptidergic signaling. So these are like small peptide molecules that they use to communicate. And the communication is incredibly complex. So like I think there are like 7,000 interactions between these neurons. So basically, I tell you, if you have this cubic millimeter of mouse brain, this is just the beginning. So you better get working, because you will have a lot of work to do, because that's kind of the essence that we're just getting into, which is totally fascinating. Which gets me now to the next question is, where in your own work do you encounter interdisciplinary perspectives? And that's, I think, really an important part. Who wants to answer that question, the interdisciplinary aspects of neuroscience? Our neurologist, go for it.
So I guess I should have said as part of my introduction that I'm going to do a neuro-oncology fellowship this coming year at the Alfred Brain Tumor Center, which is here at UW. And so in a very literal sense, pretty much any patient with cancer has to have an oncologist. So if it's brain cancer, you need a neuro-oncologist, a neurosurgeon, a radiation oncologist, and then behind the scenes there's a neuropathologist and a neuroradiologist. And if you practice neuro-oncology, you have to talk to all these people every single week. Not to mention all the social workers, psychologists, and nurses, and the other people who sort of help your patients access the care that they need. So that's something that I like a lot about neuro-oncology, and one of the reasons that I wanted to go into it is the ability to work with people from other disciplines. Sometimes I tell patients, the good thing is that there's more and more knowledge out there, as people have just been saying, and the bad thing is that each person can only know so much knowledge. So the fields get smaller and smaller, and I think it really helps to talk to other experts, because I just can't know everything. And then I think on a sort of more abstract level, something I just like about medicine, and I guess about neurology in particular, is that you have to spend quite a long time talking to each patient to understand what it is they're trying to tell you. because they're going to have some very strange perceptual experience. They're going to say, I can't see, and then you say, is it just one eye or is it both eyes? How can you not see? Is it blurry? Is it double? Is it black? Is it the colors are not right? So you just spend a long time just face-to-face with people, and you get to learn a lot about their lives. And I think it's cool to ask people what they do for a living, because I don't understand any job that's not mine. And often that does intersect with whatever problem they've come with. And I like that interplay between how people's health affects their lives.
Fascinating. You know, you might wonder how the life of a neuroscientist look like, you know, like when you become a PI or a professor or something. And really, the life of a PI is all about interdisciplinary interactions. So basically, you have, as a PI, a lot of postdocs or graduate students or undergraduate students, and each one of them has a different background. And so I always compare it like running a little orchestra of amazing, amazing people. And so like if you have a first-class orchestra, you know, you expect you to have the best violin player, you have the best cellist, you have the trombone player, and everybody kind of specializes into something, and you wouldn't mess with the expertise of a violinist. You would really try to bring this all as a team together to advance science. And so the beautiful thing about being a neuroscientist is you always work with these amazing, intelligent people. So everybody in my lab is 10 times smarter than me when it comes to math, or when it comes to genetics, or when it comes to engineering. And so basically, neuroscience lives from interdisciplinary research, and everybody sitting here in the audience brings something special to the team, and that's what we need to solve these problems. So I think that's what I think is incredible. So the question we have here is, what are some of your favorite intersections between neuroscience and other fields? Why do you find them fascinating? Andrew?
Fascinating.
Yeah.
Yeah, I was just thinking, looking on the panel here, so I work closely with the Albert Brain Tumor Center, so, you know, I'm sending folks over there after you take a brain tumor out. I was in Grid Lab when I was a resident. Jeff was my PI. We send brain samples to the Allen Institute from our brain surgeries. When we take out brain, we'll, you know, reserve a little piece and send it over, and then I see you at Grand Rounds every Wednesday, so, you know, figure a way to work you into it too. But that's, you know, in terms of some of the favorite intersections, it's kind of the things that are surprising that you run into. Again, sort of strange coincidences. One of my colleagues, his sister, is in the bioethics group, and I was a philosophy major, and she wanted to write a paper about how we started with looking at personal identity after certain types of brain surgery. So she wanted to say, okay, well, you treat epilepsy, somebody has seizures that don't stop, and you take out a temporal lobe and you cure them. What happens to that person's identity? Does it change? Are they the same person? What are the criteria you look at? And it's kind of like a throwback to stuff that I was doing when I was your age, and I was like, oh, this is kind of nice. I have a completely different perspective on it. Before, it was very abstract, very like, oh yeah, this is a nice theory, this is a nice theory, this is a nice theory. And then you see some folks that you've operated on, and you know, both good and bad, you see, you know, people who, you know, I tell folks, I can tell when I walk in if somebody's seizure-free afterwards because they just look different. But they're still the same person. You know, you wouldn't say that they're not kind of continuing the story of their life that they've started. And so that was kind of just a nice little surprise working on that paper. We've had a couple other follow-up papers, and it's in a completely different field than, you know, doesn't help my CV all that much, you know, just in general. But I think that's kind of one of the things that's interesting, is it does touch on everything. Even if you're, well, I guess if you're working in this field, you can reach out and do other things. And, you know, in the end, one of the things that's always fascinated me is, like, why are we doing all of this? Like, what are you doing it for? I think there's, you know, you can describe things at a molecular level, and that's there, but it's going to be used for something. And thinking about that and how we go about doing it, how can we, you know, address some of the ills that we all see in society versus, you know, very concrete illnesses. I think that spread is really kind of the most interesting part of what I get to do. And then again, to kind of stress the point, I don't have time to do any of this stuff, right? And so you always need somebody to work with. It's important how you connect to folks. So you don't get to just drop that part of it. Once you get to a certain point, you actually build on it more and more as you go through. And so, yeah, again, just kind of looking at people, I'm like, okay, maybe I'm doing okay with like with the different parts of this field that I'm involved in. But overall, I think that's something to kind of keep in mind while you're doing this, is that there's a purpose to what you're doing, and kind of how you interpret facts is always colored by that. And so on one hand, it's good. On the other hand, you have to be careful about it. So I just find all of that interesting at the meta level too.
Can I jump in real quick, Nino? Yeah, so just to build a little bit on what Andrew was saying, actually, I work with Andrew's patients, and I couldn't actually, some of Andrew's patients, and I couldn't do my job without being able to collaborate with all of the amazing neurosurgeons in the area. And it's, yeah, it feels like a very strange but also incredible opportunity to be able to go in and take, the situation because I work in the epilepsy monitoring unit. That's where I go to collect data and work with patients and do some experiments if they're up for it. But to be able to take the situation that, of course, no one really wants to be in, but then hopefully use those data as best as we can to learn something about the brain. If we're able to collaborate, I'm not an epileptologist, I am not a neurologist, I can't really help on that side of things, but I can learn a little bit maybe about how the brain develops and how activity changes as different parts of the brain are maturing or as different structures in the brain are changing, and use some of that more basic data to hopefully someday down the road inform more about how we think about these diseases, how we think about different neurodevelopmental processes and how those could lead to changes in brain function. And I also have the opportunity to hopefully make those data accessible to other people who are interested in maybe tackling more of the direct epilepsy types of questions and learning a little bit more about different subtypes as we go in and collect more and more and more data and start building up databases that give the opportunity to ask these larger questions. And so all of that is because we as neuroscientists like to really have this interdisciplinary approach. We like to work with people. We kind of was already said, we need to work with people because things are so complicated. There's so much we don't know. It's a relatively young field, and getting that diversity of perspectives is something that is what really helps drive things forward. And so it's very rewarding in that way, especially here at the University of Washington and in the greater Seattle area. There's so much neuroscience happening. There are so many experts and people who just are world-class scientists and leaders in the field. And so to be in that environment and be able to work with them is just, it's quite the experience and something that's very, very fun, very exciting to contribute to.
Wonderful. You see, basically, that's a beautiful example for how neuroscience works. So you have Andrew, who is a functional neurosurgeon, then Jesse, who is a PhD graduate student, or postdoc, and but comes from the basic sciences, and they work together to solve neuroscience problems. And I think what you're seeing in the field is more and more you go from, let's say Drosophila to mouse to human. So now you can do a lot of things to understand human function that were really totally out of reach. And Andrew is specifically a functional neurosurgeon, so they're involved in basically replacing functions through electrical stimulation. For example, in Parkinson patient, you've seen the substantia nigra, this little black thing in the brain. You stimulate the subthalamic nucleus, and then you can stop the tremor. But functional neurosciences is really just at the beginning in a way, because at the moment they use often electrodes that are pretty huge. I bet you in 20 years they will say, "Can you imagine?" But these days you can do optogenetics and chemogenetics, and I guess neurosurgeons in 20, 30 years will use viral vectors to be more specific. But I've seen amazing successes, like treating depressions, treating OCD, treating other affective disorders. So I think it's just the beginning of functional neuroscience. Why don't you continue, Andrew, because you know better.
Yeah, no, I mean, again, this is, I mean, if you let me, I could sit here and talk for an hour about all the different cool things we can do, and then just my personality, I'll just moan about all the stuff we can't do. And so, innovations in neuroscience, people are excited about recording from single cells. We've been doing that since the 1950s, but 120 single cells at the same time in a clinical clinical use electrode that we could, we're not quite doing it yet, but that is exciting to me. But again, part of this is just, the electrode we implant for treating Parkinson's, it's 1.27 millimeters wide and there's 8 contacts on it, right? And if you think about that, that's millions of cells and we're just kind of blasting them with stimulation. It's pretty crude. Again, they've been doing that since the 50s and 60s. We're better at it, but it's still like, when are we gonna get to the good stuff? But the thing about it is it works really well. You have somebody who's got Parkinson's, you start medications. I'm sure folks in the audience have seen people with this, but initially you take meds and you're like, oh, I'm almost normal. As long as I remember to take my pills. And then you see that kind of the effects start to wane a little bit, start to have side effects from the medications. And then you can do the surgery and you take people back 5 years. And it's an extremely gratifying experience from my standpoint. But at the same time, you're kind of in the back of your head, like, wow, we are not dealing with any of the actual problem here. We discovered this by accident. Another neurosurgeon in the 60s gave somebody a stroke and found out it fixed their tremor, and we've been kind of duplicating that effect with electrical stimulation since then. So I agree that we're not the ones figuring out why it worked. It's working with neuroscientists, neurologists. Again, in all of the surgeries that I do from the functional side, we're doing multidisciplinary clinics beforehand. We see physical therapists, neuropsychologists, neurologists, and kind of going through, electrophysiology for epilepsy. We're looking at EEGs and all of that. And again, you can't really ever do that on your own. But that is, again, one of the great things is in the end, again, what is this for? Well, okay, it's easy when you're trying to make somebody better. A lot of this stuff we're looking at now, kind of in the back of your head, you're always thinking like, well, can we make everybody better in some way? And that's something to kind of definitely think about. Yeah, it's one of the great parts about this is that kind of everybody gets involved.
So the next question, can we go to the next question? Okay, so the next question was. Can I say one thing about I just want to talk about why neuroscience is like a special space for interdisciplinary work. Like, it's the only part of biology that we can't reduce. We have to study it in its intact form. As Andrew is talking about, we studied, single cells and their spiking behavior, extracted from their context, and that seems to be not at all a good representation of what's happening in the brain, because the brain is a huge amount of different neurons interacting at once throughout the lifetime. The environment is changing. You have to bring interdisciplinary methods to make any progress in this field, because you have to study small things and big things at the same time in intact brains. So.
Yeah, I was a little hesitant to speak up because like the cell types are my thing, and so like that. But for me, I feel like that was something surprising for us when we started to do these, sequence, millions of cells and get their full gene expression profile. Then we started to see, just based on gene expression, how many different cell types there are in the brain. Like we have been studying cortex for a long time, and that's the part of the brain that's closest to the skull and is thought to be involved in higher order signaling. And most people thought that the excitatory neurons in the cortex, that there was just like a few types, and then we started to look with these transcriptomic studies, and then now we see that there's, based on transcriptomics, there's hundreds, and then now looking out at other types of properties like the anatomy and their functional properties, like we see that's largely supported, and so there's way more diversity than we anticipated, and I think that makes sense with the complex set of behaviors that we have to do. So. Yeah, and now we are going to know their full synaptome that goes along with that diversity of cell types. So it's pretty exciting.
It didn't work. I thought they took my microphone because I was talking too much, but maybe it wasn't the reason. But anyway, so the next question we had, Staci, thanks so much. You're speaking from my heart. So the next question was, and Kyobi already addressed this, how does working with the brain at the microscopic level via, for example, molecular biology and EFIS compared to looking at the macroscopic level? And in fact, going over to the ethical level and the society level, because of course, understanding the brain has a lot more implication than just understanding neurons. It's all about emotions, how you act in society, etc. And one of the big questions that we often ask ourselves is, let's say we find a gene therapy that allows someone who has never been able to speak to suddenly speak. What's what are we doing to this child that suddenly has a communication that the child didn't grow up? Or we have seen this also with suddenly people that were blind suddenly can see something. And in many cases, these people actually did suicide because they couldn't handle this. So basically, a lot of the question that we have to address is, and I want to say it maybe too non-professional, but if we mess with the brain, what are the consequences for that? And we have really no clue. So we have to work with ethicists and we have to work with psychologists in order to really say, what can we do? And what is actually a neurological disorder? What is a psychiatric disorder? Like autism, for example, is a strength also. It's not only a weakness. So basically, a lot of these questions, I think, are much bigger than solving the genetic underlying or underpinning of a neurological disorder, for example. I don't know. I'm sure you want to say something to that. Yeah.
You very briefly in the previous question mentioned neuro-ICU monitoring, and that I think is a really interesting place where, because a lot of times I think one of the things that I liked first about neuroscience and neurology is that these feel like pretty different skill sets to me, like learning how to do electrophysiology or molecular studies versus thinking about these like very broad, abstract, like ethical, social questions, like Dr. Crow was saying, like, what is consciousness? Like, what does it mean to be the same person as you were before? Like, what are those criteria? What does that even mean? And so sometimes those can feel like quite separate skill sets. But one place that I think it's interesting to think about them coming together is in the neuro-ICU. Sometimes you have these comatose patients, and I'll say before very recently, actually all the time, the general principle with comatose patients is that you would rather keep someone alive who has no chances of recovery than accidentally disconnect someone who does have chances of recovery. This is always sort of like the principle by which we make all these rules as to what does brain death mean? Like when we say someone is brain dead, there are criteria for that. Like I've declared someone brain dead before. And now those criteria being complicated by relatively recent findings that there are EEG signals from those people that you wouldn't expect from a truly brain dead person, meaning there's no activity in the cerebral cortex and there's only like brainstem and below or less. And that has obvious ethical implications. And then there's questions about like disability justice, like what is a good life? Are we in a position to decide that for people? You ask their families, and then later there's a question about like medical communication. That's a whole separate conversation. How do you communicate that information? But that was an interesting topic for you to have brought up, the role of EEG monitoring in neuro ICU, because that's like a place where the micro and the macro aspects of neurology really come together.
In fact, it really gets to this whole question, how is the brain organized, and how much do you really understand? And I talk to neurocritical care people where, you know, there are children that are born without a cortex. And also basically, theoretically, they can't process complex things. But when you talk to the parents, they say, we can communicate with this child. You know, and then often doctors say, hey, but this child doesn't have a brain, kind of, you know, but we don't understand it yet. You know, there are so many forms of communications that we only look for the most obvious communications, but there are many, many different forms. And so we're just touching this whole question. And I just want to, one experiment I've seen once, people remove the cortex of a cat, and the cat was walking around through a labyrinth, and you did not see any difference from one that had a cortex. And then there are all these experiments of learning. You take away the cortex of a mouse, they still can learn very much, very quickly. So there's a lot of communication and information processing in the brain that we don't understand. And I think the clinicians and the basic sciences have to be very humble about this whole brain and thinking we understand it. Jesse?
Well, yeah, I think we need to be humble from a scientific perspective, but also as neuroscientists from like a lived experience perspective too. We need to, especially, you know, when we're working with patient populations, or, you know, anytime we have contact with other people, or our work is going to potentially be touching on someone's lives, it's very important to understand the ramifications that has from the perspective of the people who you actually want to be supposedly helping, or hopefully helping. And I think that's something that neuroscience is still grappling with and figuring out, and how to get better. I work with some people at Children's who study autism, and that's one field where I think that's been like a really good example of how to bring community members into the work, and how to discuss, you know, with and hear from that population about what is important to you. What is it? What do you think of or hear when people are talking about neuroscience? And just maintaining that open communication is definitely something that needs to be an important part of the work moving forward when we move towards away from these, you know, very basic science questions, or even when we're thinking about basic science questions that have a clear, you know, clinical or therapeutic outcome. Because we often just don't necessarily know even the right questions to answer, or what would be the most useful way to go about answering them for someone who has a different lived experience than we do. And so I think, you know, that's this, again, getting at this very macroscopic and ethical level. It's definitely an area where we could use some improvement, and also need to just keep thinking of as we're going through our work as neuroscientists.
Yeah, thank you very much, Jesse. So Jesse basically led us also to the next question, because what's the role of communication in science in general? And you know, like we talked about communication with patients, but also among the scientists, communication plays a huge role. So just, I tell you, like, I have a lot of ideas, and everybody here has a lot of ideas. And especially at night, these ideas come, and you think, wow, now this is the experiment I want to do. But you have to convince reviewers, you have to publish it somehow. And so the trick of communication is really like trying to express your thoughts so another person understands this, because that's kind of what's critical, not only for the community of scientists, but also community of the public awareness, you know, like how can we communicate our ideas? Why is it important to have a single cell identity? And why is it important to invest in science? This is all very, very important questions that I think we all as a scientist maybe have to learn to get to do better. Kyobi, you're a teacher, a lot of lectures, and you know.
Yeah, like I think that if you went outside and asked somebody, how does your vision work or how does your hearing work, the probability that they would give you the right answer is like probably approximately 0, right? Probably rounds to 0. And I think that that's like a huge failing on science overall. And it's not because us as individuals aren't interested in doing that kind of communication. It's that it's really hard. It's really hard to communicate really complicated things to people. But we have to try more. I think that it's like really important. It's on all of you now. You now are the science explainers to all of your family. You have to be able to tell them how these things work and to teach them how their brain functions. So I would just make the plug that that gap is really, really big, but I don't, but also this communication amongst each other is also very important.
Yeah. Andrew.
Yeah, I would just add to that. I think that that's, you know, kind of what I was thinking about when we're talking about communication in science. And say what you will about current state of affairs in the world, but part of that is, you know, it's our fault as you know, having the being sort of we talk to the same people all the time, right, about science stuff. I can use certain words. I'm going to talk to Dr. Chee, and we're going to throw medical terms around. And obviously, when we see patients in clinic, I'm generally trying to gauge, like, okay, how should I, you know, phrase things? What kind of language can I use to make things more clear? If nothing else, to get like a general overview of an understanding of what we're doing. And if I'm putting an electrode in STN and I tell that to somebody, I'm going to put a Lexel frame on, we're going to use coordinate, you know, the Lexel coordinate system to whatever, they're not going to know. I think we've kind of, all humanity in general, I guess I could say, has done like a bad job of doing that, trying to, you know, see where the other person's coming from, giving everybody a little bit of grace, and being able to communicate the ideas in a way that's not going to immediately alienate people, right? So I just wanted to add that it's not, you know, from the science teaching standpoint, yeah, obviously we want to get people interested. You have to do that in a certain way and be accurate. But it extends to other stuff that we do is yeah, you want to maintain accuracy, but you know, you have to be careful about injecting your own value system into what you're saying. That, you know, we like to think, oh yeah, this is right, this is capital T truth, and there really isn't any such thing, right? I mean, we can characterize every connection of every single cell in the brain at the human level, and okay, well, this is how the brain works, and then I'm going to stick an electrode in it, and it's all going to go to crap, right? So there isn't like an unchanging, this is the way things are, but we all want that, right? We like, again, this is how our brain works. I'm going to immediately like counteract what I just said, but there's this natural tendency to be like, hey, this is, you know, how things are, and it's not like that. I mean, there's things you talk about, innovations, changes in the field, all of this stuff. They're all conditions of possibility for thinking about this. If you ask somebody from the 1600s about brain function, you know, there's certain things you're never gonna be able to explain to them. And so all this stuff does change over time, and we kind of have to acknowledge that and still try to navigate, again, what are we doing this for, right? Try to figure out ways to kind of bridge the gaps that we come to, so.
Yes, Michelle.
Can I actually ask you a question? It might involve dating yourself, but I graduated from med school in 2020, and I was under the impression, in my school we had like specific directed classes about how to talk to patients, like how to run a family meeting and how to elicit a sexual history in a sensitive way. Did you have that? Like, I don't know when they started that.
Yeah, I'm not that old.
Okay. Sorry.
No, I mean, and I guess this is the thing that's funny about it, right? I mean, we've been doing it for however many years. As soon as I finished med school, started residency, wow, all that stuff was kind of helpful, but also really not that helpful. And then, you know, when you're a trainee, there's a certain way you address it. And then as I'm sure you know, as you switch to being an attending, all of a sudden it's a very different perspective because then, oh, there's nobody else to ask. What I say goes. And so there's another shift. I mean, I think like the Dunning-Kruger thing is very true. We're trying to teach people, oh, this is how you talk to families. And it's at the point in their like knowledge development and experience that it's kind of going to slide right off, right? Smooth brain, no wrinkles. I mean, it's a hard problem. I'm not saying I have the solution, but yeah, I would say like all this stuff that I thought about and I learned, oh, this is how you properly do it. You run into like counterexamples as soon as you start, right? Oh, they're from a different, you know, plenty of examples of me trying to kind of follow that script and basically you're digging yourself a hole with this family who's like, oh, this person doesn't know what they're talking about because they keep asking me what I'm thinking. Yeah. You know, like the patient's like, no, why don't you just tell me? Yeah. And you're like, okay, well, I don't love to do that, but if in the end that's what you need to do, I mean, you can still phrase it and kind of give them a choice, but you kind of have to figure out what they're looking for. And it just takes time, right? And so, but yes, I did have that training. Not that it was useless, but it was very, again, it's kind of like a step on the way, right? And again, I'm sure at some point, maybe I reached that point today when somebody's like, did you actually learn how to do this? Because I'm dated and I'm a dinosaur and I think I see things in a certain way. I think you kind of have to be comfortable with that and kind of accept like, okay, I've been doing this for a while. Maybe there's stuff to learn from how society is now, right? We think about things definitely differently than we used to, but and I kind of keeping that open mind and being, like I said, having some grace when you're talking to people, like trying to be humble and like, look, I don't know everything. But at the same time, you know, I have been around a little bit longer. If a resident's talking to a patient in a way that I'm like, oh, that's probably not the best, I will tell them. And yeah, you can say like, what's the best way to do it? And obviously we debate that, but yeah, it's a process for all of this stuff, right?
Yeah, for sure.
Staci, you.
I just wanted to say something really quickly that I feel like as scientists, like I actually never learned how to talk about science in a way that was, I feel like, easy for other people to understand. Like I tend to just go deep into the details and get caught up in like the specific thing that I'm studying. And I feel like, you know, at this point in my life, it's, you know, a real loss to have not spent time making sure that I could talk to my kids or making sure that, you know, like my grandparents were excited about what I was doing and had some sense of understanding of it. And now, you know, now I'm working on it. I'm still have a long ways to go, but I feel like we don't have a class like that. We need that class.
No, but you learn it. I mean, like the thing is, when we talk about interdisciplinary research, it's all about communication between different disciplines. Like I have mathematicians that can only talk about bifurcations, and I have people that only talk about reactive oxygen species. I have people that only talk about the plasmid and whatever RNA interactions. So basically, the trick is you learn to communicate with many, many different people in order to get most out of it. But I think for me, the most fascinating communications are often with parents and patients. You know, like as a basic scientist, you think you understand things like schizophrenia or epilepsy or something, but you get a totally different perspective when you talk to the parents and the patients. You know, you suddenly realize, oh, this is important for them. I thought this is important, and so I think working as a basic scientist like Jesse in a clinical setting is incredibly eye-opening, and I think very important where we want to go. And I think, as I said, when you become neuroscientists, I think there will be a lot of fun for you to be interdisciplinary because you can get a lot out of it. So, Kyobi, yes, please.
On one last thing, like as Andrew was saying, at the time you're in the clinic, now you're trying to teach the person in front of you because they don't really have a strong understanding of the scientific ideas, right? So this is the call to action. You are the learned few. Try to teach. It's okay to be wrong. It's okay to edit yourself and work and continue to improve, explain things to people, teach. Our whole society needs to understand what science is and what science does.
So, my God, there are like 100 questions that we haven't even started yet. But we want to give you also a chance to ask us questions. So maybe the last question before we go into intermission and then give it over to you is, what was your path to neuroscience? How did you decide to pursue a neuroscience career? Who wants to start?
I might have the freshest memory on this, so I'll go first. I mean, it's also somewhat of an easy answer for me because gray matters was a big, huge part of determining how I went into a neuroscience career. A student just put out a call that I ended up seeing from like a listserv at UW, and wanted as many people as she could find to join this first meeting, and we had like maybe 15 or 20 of us that stuck around for a few months, and I got to hear about people doing research, which I honestly didn't even know you could do at UW when I got here, because no one in my family had been to college before, so it was very just like a lot to process, and it was really fun. It was cool to meet all these other people who were so excited about the brain and had so much more understanding of it than I had, even though, looking back, obviously they were just starting their careers. They had like 3 more classes than I had. But it was just a lot of fun. And it really stuck with me and encouraged me to go join a lab and learn a little bit more about what the hands-on experience was like. I remember like sitting in a basement one day doing some what's called fluorescent imaging of the brain, where you can see neurons lighting up and individual cells being active and kind of blinking on and off, and it it looks really kind of like almost like a night sky, and it was this really just kind of incredible experience to think that, like, if I do this long enough, I might see something that no one else has ever seen before. And be like, that blew my mind. Like, I couldn't believe that was even, something that I could even imagine being in a position to do something like that. And so it stuck with me for a long time. And I think neuroscience in particular, there's so many opportunities to have that kind of, quote unquote thrill of discovery, because there's so much, like we've been talking about, to still learn and still understand. And so I think being in that kind of unique position in this field at this moment where there's so much there's these new technologies, there's this real push towards interdisciplinary work, there's this push towards open science and collaboration. It's just makes that thrill maybe a little bit easier to come by too. So that's where I come at it from.
Jesse, that's so wonderful, because I feel like everybody here had this blow my mind feeling of suddenly, wow, this is what I want to do for the rest of your life. And I think as a student, it will be very fascinating for you, because often you don't know what will blow your mind, and also there are many different things that can blow your mind, like some people get totally excited by an ion channel, others get totally excited by connectivity, others by the psychological implication of neuroscience. So you have to wait until it blows your mind, and you have to be also prepared that it might be unexpected. Like, I never wanted to be a neuroscientist. I wanted to be an ecologist, and also I never wanted to be anything like a clinician. And then suddenly I stuck an electrode into the brain, and the brain talked to me, and I thought, my God, what a privilege to study this holy thing. And it was like blowing my mind, as Jesse said. And then I was stuck. It never went away. So one more person or 2 more person? Jesse? Michelle?
it's great that people want to do research. I don't really. A lot of you probably feel like you have to do research, which is probably true if you want to go to med school, which is annoying. I think it's quite a different skill set than being a clinician. I think it's very interesting to listen to research. I don't have the patience to keep doing it enough times until I'm the first one to find out something. I just don't have that much patience. But I had an actual answer to this question, which was, why did I want to do neuroscience? I read a book by Oliver Sacks, which is called I read Awakenings first. His most famous book is probably The Man Who Mistook His Wife for a Hat. But Awakenings is a series of vignettes about 20 different patients with encephalitis lethargica, which is also known as sleeping sickness. And it's about the advent of L-DOPA, which was a drug that was new at the time, I want to say the 1960s or 70s. At the beginning of the story, all the patients get this drug, and it's like a miracle, and they all wake up. They've been institutionalized for like decades, and they wake up, and it's like, wow, they're gonna go home, they're gonna be cured, like this is just a miracle, and then, et cetera, complications ensue. But each of the patients has a different physiological and personality reaction to the drug, and they each come to a decision with their doctors in a different way by the end of their vignette. And so it was maybe from that more like one person at a time perspective that I got interested in neurology, and just that interplay between physiology and individual people's personality.
See, it's so funny, Michelle. I work so often with clinicians, and clinicians are different than neuroscientists often. And I had clinicians working in my lab, and each time they recorded from a neuron, they asked the neuron, what do you tell me about the brain of a pediatric patient? And so she was like totally asking these questions, but seriously, you know, like, how can I translate this into the clinic? Which is really telling you, you know, like how you have to find, what's your passion? Is it the clinic? Is it the neuroscience? Is it engineering? Is it molecular biology? And then you go after that. And I think neuroscience is a field that welcomes everybody with a different background. The broader the background there is, the better it is for the sciences. So, yeah. So we have Kyobi.
I think it would be remiss for me not to address this question, because I probably have the most unusual background, I would say. So when I was 18 years old, I was a high school dropout. I was playing video games all the time. I then became a plumber for 3 years. The chance of me being here is like, again, approximates to zero. The moment of discovery for me was actually when I went to college and realized that we could rationally understand the world. Like before that, for some reason, school was just was a drag. Like I just did not want to do it. I didn't care about what anyone was saying to me. But when I went back to college, I realized that like there is a set of approaches that you can take to understand, and at least to the best that we can. Of course, these truths are always changing, as Andrew was saying, but at least to a first approximation that you can use techniques to understand the world. And I think that was so powerful that it really awoke a drive in me that lasted, you know, the last 20 years. So yeah, high school dropout, plumber, got a GED, went to undergrad at Evergreen State College, no grades, woohoo. Came up here, worked at the Hutch, went out to Harvard for a PhD, and postdoc, and came back here. So it's a very unusual pathway. So anyone can get here.
It's not so unusual. I have a very, very good friend, famous professor at UPenn. He lived for like 10 years without water, without electricity in a cabin in Colorado before he decided, oh, maybe I should study the brain, you know? So it's not something totally out of the world. It happens. So, okay, we have an intermission and we come back. So ask us all the tough questions and we're ready to answer them, okay? So we have 10 minutes, correct? 10 minutes?
It'll be 5 minutes, but I'll
What?
give a little speech. It'll be 5 minutes, but
5 minute break.
Yes.
Okay, thank you.
before we all break, let's give a huge round of applause to our amazing panelists. And as Dr. Ramirez said, we'll be taking a 5-minute break, and then you guys will be able to come back and ask the panelists your questions. As I mentioned before, Gray Matters is an organization entirely run by student volunteers. Thus, we heavily rely on donations to make all of our events like this one possible. While you are enjoying your break, please consider scanning the QR code at left and donating to our club. Furthermore, please make sure you have filled out the RSVP form to make sure that you're entered into our raffle, and the first prize will be announced when our break comes back. See you in 5 minutes. Wait for Sarah, Because Sarah has to say this next thing. Sarah.
No, I'm just gonna say it. Could everyone? If everyone could start finding their seats again, it'd be great. If everyone could start finding their seats, we will have time at the very end of the event to come back up on stage and ask the panelists questions. But for now, we would really like if you could take your seats in the audience, and panelists, please take your seats again as well. Thank you.
I'm sorry, I was just not there. Okay, welcome back everybody. Please begin finding your seats again. Before we resume our event, we have drawn a winner for the first raffle of the evening. Congratulations to Annika F. Are you here? Okay. We have another backup winner, Torres Z. Yeah, come on down. Woo! Or actually, do we have the bus? Oh, we do, okay. Yeah, so you can collect your prize here and stop by the store table on your way out. Thank you to our generous sponsors for helping us put together our prizes for the evening. Stay tuned for another raffle drawing at the end of the night. And we're switching. All right, we will now move to the Q&A portion of our night. So there's going to be two ways that you guys can ask questions. You can either come down into the aisles where we'll have some of the EWN team members holding microphones, or you can scan this QR code and join this Slido. And if you ask a question on Slido, the rest of the group can vote on the most popular questions, so we make sure that they get asked. We will alternate between in-person questions and Slido questions. And think about what curiosities brought you here tonight. This is a great chance to ask them. We have an awesome panelist here, so take a moment and ask some questions.
So just so you know, we are now very nervous here on the panel. You know? So what is coming at us?
Just gonna leave it up for a few seconds so you guys can get your questions in. I'll stand on the side.
All right, I think we're going to start with an in-person question. So Naomi, if you want to ask the first question.
I would say the answer is the same to both. The first one and the second one have the same answer, and it's that care about people and talk to them and engage with them directly. That's how you build a network, and that's how you matter to other people.
So the brain is really a very special organ, and that's objective. Because, you know, in most other organs, it really doesn't matter what each cell does. Like, let's say you look in the skin, you have maybe 6 cell types, and you can take a piece of skin, put it somewhere else, and these 6 cell types will still do what the skin does. If you take a piece of a liver, and you understand how these 6 liver cells work, you understand pretty much what the liver is doing. It's, you know, and you go on with all these other things. And when it comes to the brain, it's very different, because now in the brain it matters what every single cell does. So if I move my finger up, I have to activate very specific nerve cells in, let's say, my motor cortex, in the cerebellum, in the brainstem, in all the other substantia nigra. So basically, the brain is very specific, because it has to be very, very specific communications, and so that makes it very unique among all the organs. And one more thing, because it's so complex, the progress to understand the brain has been also slow. You know, like you basically are now starting to ask these much bigger questions. You know, what is perception? What's consciousness? And how do I feel? And all these questions are now starting to get addressed, because we get a better understanding of single cells, how they're connected, how they communicate, etc. So I think that makes neuroscience very special.
We will be reading our first audience question. I guess our most popular Slido question right now is, if the Phineas Gage accident were to happen today, how would it be handled with modern advancements?
I guess maybe directed towards me. Huh? You know, probably not, in the end, not all that differently. I mean, you'd have to remind me again when exactly it happened, but I think it was before antibiotics. So, you know, that would probably be the major difference. I mean, we're not at the point where we're like fixing brain, right? So this thing went in here, came out, basically gave himself a little bit of a frontal lobectomy. Stuff like that happens all the time now. We have better supportive care. You know, if he hadn't been conscious, walking around, all of that, he probably wouldn't have survived. And now we're just better at getting people to the hospital, getting them in the ICU, ventilated, preventing infection, secondary damage, keeping people around longer to try to figure out if they're going to make it or not, that type of thing. But given that, you know, again, just from what I remember about what happened, he was like fine the whole way through. And so that's kind of like, we would be like, okay, well, yeah, we're, you know, debride the wound, close it, antibiotics, make sure he doesn't get a big brain abscess, and, you know, sort of secondary damage we're a lot better at handling. But in terms of trying to, hey, can we make it so he didn't, I mean, his behavior described afterwards kind of what we characterize as frontal, right? Not so executive function, not so good at planning, short-tempered. We still see that now. We sometimes do that to people on purpose if there's a good reason. You know, if they have a big frontal lobe tumor and you take it out, yeah, you prolong their life, or there's some personality changes. Sometimes yes, sometimes no. That particular part of the brain's kind of, one, it's massive. Like, if you ever seen what comes out after a frontal lobectomy, I have pictures if you want. You're like, that's like the size of my fist. Like, what? You could take that out and people are pretty much the same. So, really long answer to this short question, I guess. But yeah, it's more about kind of the other parts. And, you know, again, kind of to go back to like why the brain, that is kind of why it's so interesting, right? All the advances we made had to do with everything else, you know, no infections, making sure, all that kind of stuff. But we're not going to fix the brain, so.
Yes, Dana, if you want to go ahead with your question.
Yeah. Our next question is, what role does neuroscience play in human nature and free will?
I'll be the bad guy here. I'm sorry, could you repeat that?
What role does neuroscience play in free will and human nature?
Yeah, so the philosophy major in me wants to sort of answer this. and this is maybe again where I'm going to date myself a little bit or be crotchety, but when we ask questions about free will and human nature, those questions, usually people are asking those questions for a reason. If you think about the free will debate, I always think like, why are we talking about free will? Because a lot of it is usually tied up in how do we hold people responsible for their actions. And then when you think about that question, it's, are you doing it to further a particular agenda? So free will's, if you want to punish somebody for something, they have to have free will, right? That said, from a more practical standpoint, are we going to find the neuron for free will? I don't think that's going to be there. So if you're looking to neuroscience for answers that way, again, I'm still going to go back to like, why are we looking for that answer? And I'd probably say the same thing about human nature. It's kind of, you can, I think, discover tendencies. I mean, psychology, you don't need I guess psychology is neuroscience in a way, but we have these tendencies, but if you answer these questions, you discover them, you can describe them, but you still kind of say like, why are we doing that? So that's my thought on it. Not very neuroscience.
I really love the question, and the interesting thing as being a neuroscientist is you observe yourself a little bit different. Like when I make a decision, I think about my brain mechanism that made the decision. And often what I realize is I make a decision, I do something before I actually make the decision. So basically, I think, oh, maybe it's time to get up, but I already got up, because so a lot of the decisions you make are unconscious, and you find an excuse, a conscious excuse why you did that. So a lot of this, I think our unconscious side of our body is a huge, huge part, and we don't understand it yet.
And we'll now go back to our online question. So our top voted question now is, what is something you wish you had known before beginning your research or physician path?
I answered this question in the pre-interview that Gray Matters did with me, and finally I decided I didn't think I was gonna want to change my own trajectory or I'd be able to. I was just a very confident high schooler and college student. I was like, everything is easy for me. Being a doctor doesn't seem, other people think being a doctor is hard, but it's not gonna be hard for me. In the end, it was hard, but I still like it, so I don't think I would have changed that path. Something I would maybe tell other people is like, it's okay and it's probably good to take a break once in a while. It's a very long path if you wanna get a doctorate of any sort. And you will get too tired and burn out, so you should take a break sometimes and be nice to yourself.
A very boring answer, but I did not realize when taking so long to go through school that the whole time I was not making any money. And then you literally start your financial life much later than people who, just get a job right out of school. And so I feel like you should know that when you go into it, and then think about what you want to happen in your life and when, and then. Yeah.
I had 6 kids as an assistant professor, and we had no clue how to pay our phone bill. Should we go to pay the phone bill, or should we go grocery shopping? But I think in retrospective, the kids didn't know that we're so poor. So I think, but by the time you become a full professor, I can tell you it's fine, correct? I mean, we make enough money.
I'm going to answer a slightly different question, and what is something I'm glad I didn't know? I'm glad I didn't know how emotionally isolating it can be to do a PhD. There are times at which you are the only person that cares about what you're doing. There's no one in the world that can feel and can understand what you're what you're struggling with, and that can be incredibly emotionally isolating. So I think if I would have known that, I may have shied away from doing it, but that would have been a huge mistake because like in retrospect, it was the biggest transformation of my life and was extremely important to me. So.
Okay, so our next question is, what is your guys' perspective on the financial barriers that limit people's access to neuroscience, both in an educational sense, so accessing journal articles and things like that not provided through your university, but also in the sense of like a clinical aspect of accessing new innovations that are coming about within neuroscience, and kind of this concept of like clickbait and like making your research and what you publicize enticing to your viewers rather than focusing on the science behind it.
I'll take the access to information or publication side of things because that's something that I think a lot about. We're kind of stuck in this weird, or we're not stuck, but we've been trained in this system where you really need, for the sake of your career, to publish in places that charge this absurd amount of money for you to actually even, like, you're paying to tell people about the things that you've found out and all this work that you've done, and it's crazy. It really makes no sense. But that's how things are for some reason, or have been. And people, I think, generally are wanting to give up on that model just altogether. And so there are new outlets, or outlets that are becoming more popular, like things called preprint servers, which are not the same as journals. People can just put out their research without it having been reviewed by other people in the scientific community, which that review process is extremely important most of the time. But those are free. Anyone can access those. And there are a lot of journals now where that's actually kind of encouraged, that you can just put something on a preprint server that's, the latest and greatest that you've been working on, and then you can just roll that into a submission to a journal. And so I think there's been a lot of momentum and more researchers doing that, and more researchers, being very careful about taking the time to make sure that if they're updating what they find, they add that to the preprint, or they add that to make sure people have access to the best information that they can provide them. But that's only one step of the process, because not many people are going to be reading those, and they're written for a very specific audience, which is your scientific peers, and not necessarily, most people aren't writing those publications in a way that are very accessible, which is why Gray Matters is such an important thing, is because y'all are the people who are making that translation in a lot of cases, right? You will take the time to research these things, understand these things, condense them and synthesize them in a way that is tractable for you, and then for the other people who are reading your work, and then for the broader community. This is only, one way of going about this. I think having events like this is another great way of trying to disseminate information. But I think one thing that I've heard that I think would be really cool to hear more about, or see more of, is we were all just emailed, right? Like someone just was like, hey, will you come do this? And any community group can do that. Like you can just reach out to a professor. They might say no, they can't come, or they're busy, or they don't have the time for it, or whatever it is. But it's worth a try, and I've seen it work. I've worked with professors who they'll do that. They got someone, just to say, hey, can you talk to us about aging? And they came in and sat down at, I think it was the senior center, and they just talked to folks about aging and what's going on in the brain and just things like that. So I think just building more of those bridges to the community and having that be sort of an interactive process is something that I hope we do a little bit more of in the future.
Maybe because I'm the old guy here, but I'm an editor-in-chief of a journal of physiology, so it's a big journal. And for me, the publications play a huge role, because basically I see publication as an interactive process. So if you do research, while you do research, you have to think about how can I sell that story as a publication. So you basically, while you do experiment, you think, how do I publish this? And so your publication is a guide in your work, because you don't want to do like one experiment forever, which actually doesn't tell a story that you can publish. So I feel like publications are really an important part, and then it's all about the truth. So you want to have a peer-reviewed process in order to really get closer to, like, very rigorous about how did you design your experiment? Was it really, did you interpret it the right way, etc. So I think this rigidness avoids that the situation that we have in media where everybody can tell you something that might not be true. So I think it's a gatekeeper of scientific knowledge that has to be there in order to prevent it from allowing you to say something that is not true. You know, I think, and we still are wrong often. We interpret our data often wrong, but as long as the data are real, our interpretation can be wrong and we can revise it. You know?
I just very quickly want to address your question about clickbait versus publication, and I think it's a very, it's a very interesting tension, because as scientists, like you have to sell your work to everyone, because you have to show why it matters that tax dollars and our attention and stuff are going into that question, right? But there's, of course, this line that starts to be, what the New York Times interprets that study as, right? So like that clickbait often comes from editors and titles at those institutions. If you read those stories, they're often very good, and the scientist is not overselling their work. And like that clickbait thing, I really don't think that's about scientists. I guess that's what I'd say.
All right, now we'll go back to our Slido question. So one just popped to the top. As a non-neuro student, brain-related work can be intimidating. Oh. How have you seen other students successfully bridge interdisciplinary work and get up to speed?
I guess I would say I was a philosophy major. I guess that's, the type of philosophy I was interested in. As an undergrad, I was like a big fan of Foucault, so this is like the farthest thing from brain that you could get. I think, the thing to do with that sort of transition is just be open to the fact that it's going to take a while. And I think finding the connections to people who can maybe give you a little bit of hand with stuff, I think it's a lot easier these days. I mean, you can YouTube anything, right? Personally, I do a little bit of coding now just for my own, like my research career is kind of like for fun at this point, right? But all the kids in lab are using Python. I'm like, I don't know, I was a MATLAB guy from college, and like, how do I learn how to do this? Oh, okay, actually some, very attractive young guy is gonna tell me how to do this with great enthusiasm and like lots of examples and graphics and all this stuff. I think it's just kind of that willingness to say like, yeah, I do kind of really wanna do this. It's nice to have sort of concrete goals that, like, okay, I'm working with this person about this particular question. Yeah, maybe it's not the most interesting thing. It's not the aha, like, oh my God, I so want to solve this problem thing. But you see it as like, okay, well, I'm going to learn how to use the Fourier transform to tell if high gamma changes are in the brain are correlating with movement or with this particular behavior. Okay, it's a very specific question. It's kind of boring. You're not going to find a whole lot of people, who want to talk about it necessarily, but like, okay, we're using this as an example for how to do that. And then I think you actually do have an advantage in some ways if you don't start in the field, because you're a little bit more open to kind of what does that mean, or like, well, one, how would I write about it? Here I'm thinking maybe it's the humanities person asking about it, but I think that advantage in communication can actually really be, that can be a really big advantage if you can kind of convey the ideas in a way that, as you were learning it, you figured out like, oh, okay, well, this helped me. I can kind of use that to bridge the gap for other people. It's actually quite helpful. I hope that's kind of what the question was about, but you know, the kind of bottom line is, yeah, you sort of have to decide to do it, and you do kind of have to look out in terms of finding folks. But I think if you look around enough, that's kind of what you. I think if you persevere, you'll generally find somebody who, you know, appreciates that you have this interest and that you're working and you're learning how to do it.
Kyobi, you want to say something? Right?
Yeah, since this is a teaching question, I do want to just address it. So, two things. First, neuroscience is the most ocean-like of the disciplines. There's so many things that you don't know, and there's so much different interdisciplinary stuff that you need to understand in order to fully get it, right? So even if you are trying to do neuro as an undergrad, you're still going to feel way overwhelmed and intimidated by all the stuff. And in fact, you might feel even more intimidated because you're supposed to know, right? And then I think the more important thing is the sooner you get comfortable with the fact that nobody knows, that your PI does not know that much either, and that we're all just trying to figure it out every day, like, just get comfortable. It's okay. It's okay to not know.
Yeah, never feel intimidated, actually, you know, like, because you have to enjoy working with people that know something better than you, because you will always find people that know something better than you, and you have to kind of profit from that and not feel intimidated. And there's room for everybody and every kind of type of person. You know, there are persons that love to go very, very specific, and there are other people that love complexity, and there's space for everybody. So don't be intimidated.
All right, we'll take our next in-person question over in Yeston's line.
This is a question specifically for Jesse, but everyone feel free to answer as well. Jesse, you are co-founder of Gray Matters, where you are an editor and writer for the inaugural issue, along with various other accomplishments. What does it mean for you to be a panelist here tonight?
That's a good question. That's a tough question, and I'll try not to cry or get too sappy. But it's a very big deal. You know, even I think at the first evening with neuroscience, like, I didn't really think I would ever be up here as a panelist. It's just like a fleeting thought that you might have every once in a while, or that I was like, oh, well, that'd be kind of cool. But yeah, it was still kind of a shock when Sarah asked if I wanted to join. So. Yeah, it's a very big full circle moment for me, and I'm mostly just pleased that it still exists and that it's been going for over a decade now, and that everyone who takes the reins has new ideas and new directions that they want to pull things in, and watching it change and thrive in the way that it has been very rewarding, and it's been a big academic impact for me too. You know, just being able, like, you know, Dr. Ramirez was saying, being able to work with people who are smarter than you and who push you in these new directions and who can do things that you can't was something that I felt like Gray Matters really exposed me to, and just kind of set the tone for the rest of my career, just seeking out those people who I thought would help me to become a better scientist, a better communicator, whatever it might be. And yeah, I mean, I'm happy. I'm happy to be here. It's a cool experience. So thanks for asking.
And we're back to our online question. How do you see the development of AI getting used in the field of neuroscience?
Big deal, you know, and I tell you why. One reason, for example, to analyze data, they are getting more and more complex, and you need to write code. You know, like you don't get commercial software anymore that analyzes your data, and the data become more and more complex. I mean, as we understand single cells, they become more complex. So AI helps you to analyze this. So nowadays, basically, I wouldn't say you don't have to know how to write code, but you can use AI to write code for you as a tool to get much more complex questions answered. Andrew, is this wrong, what I say?
So I would actually be kind of like an AI optimist here to say that, like, I mean, there are some, we can be all really scared or whatever, but like, I think that what AI is doing right now for neuroscience is absolutely unbelievable. Like, I think that where we're really making progress right now is matching behavior to neural signatures. And AI has basically unlocked the ability to track behavior in complex settings instead of in extremely limited, head-fixed, like very reduced settings. So I think, like, and then of course, as Nino's saying, then analyzing the data, finding structure within the data, that's all machine learning, that's all AI. Like, people are just deploying this all over the place. I think it's really exciting.
And at the beginning of this evening, you saw basically, this is the stripe of the motor cortex. This is the somatosensory cortex. That's where you feel your somatosensory impressions. Now the reality of how the brain works is far removed from that. Because basically, during a motor act, your entire freaking brain gets active. And basically, many neurons get synced to this motor activity. So to understand this complexity, how these different areas work together to generate a motor movement is just like unbelievably complex. And AI starts to help you by writing, you create these so-called manifolds where you can see how this activity spreads during different areas. So I think we're getting a fundamental different understanding, which a human alone wouldn't be able to do.
Just to demystify it a little bit, AI is not magic, it's just fancy correlation. It's just correlations, but in a way that we couldn't understand or like really appreciate them before. So it's just the same thing that's been happening for a long time.
And just as like a little counterweight, because I have used very sophisticated machine learning softwares for doing data analysis and stuff, but one thing I will never use AI for is interpretation. I think that should be in the domain of the experts. And that is definitely like what our job is supposed to be, is in understanding these things that are at kind of a level where, you shouldn't expect something that's just kind of picking up on and regurgitating things that people have already thought of and said for a long time. That it can't go really beyond that, or some people might claim that it can, but I don't trust it yet. And especially because it's built on faulty information in a lot of ways, right? Like we don't have the right answers. And so if you're putting in a bunch of information that is questionable at best, then you can't expect it to put out something that is going to do much better than you can, or that you and your field can do especially. So I think there are great uses for it, but don't rely on it for everything.
This is an interesting place. We had been talking earlier about ethical things. I think I've heard of people trying to use AI to, like, for example, read chest x-rays or something, especially in resource-limited settings. And that's always kind of an interesting ethical question, right? Like, it's probably better for people who don't have access to a radiologist to have at least an AI, but maybe we should try to get them a radiologist. And how much time should we spend in that in-between place? There's a lot of studies that we do actually in other countries. Like, for example, here in the States, the standard of care for a stroke is that if you can see a clot, we go and try to fish it out. If we want to do a study where we ask, but do you really, is it pretty good if you just give them aspirin instead? We can't do that here. We have to do that in a broad place where they don't have the capabilities to do thrombectomy, which is where you fish the clot out. And I think that raises a lot of interesting ethical questions.
All right, now we'll hear our next in-person question.
Okay, this might be a lot to throw at you guys at once, but we're gonna be combining kind of two questions here. What has studying science or neuroscience changed how you view yourself and changed your psyche, along with what have you learned about yourself throughout your journey, whichever journey you guys took, and what undergrad experience most greatly impacted you and like led you to where you are today?
Well,
I think what science did for me is instead of being a know-it-all with not knowing what I was talking about, now I'm a know-it-all that has at least some idea about what I'm talking about. I'd say that's like the big way it's affected me is like it gave me some rational backbone to actually like apply tools of thinking in order to problems. But I used to talk out my all the time. And then, wait, the other question was, undergrad experience. So my most important experience was having a professor, Betty Cutter at Evergreen State College, believe in me like for the first time that anyone had ever really believed in me. So that was extremely important.
I'm gonna have an unconventional answer. I did major in neuroscience in college. It was, I don't know, fine. The best thing that I did probably in college was that I studied, stop laughing at me, was that I studied abroad in Spain, which was a lot of fun, and also I became fluent in Spanish, and that's very helpful in a very practical clinical setting. Patients like it, and I like it. And I also just think it, you know, we had been talking about interdisciplinary stuff. It kind of broadens your mind and makes you think about other ways of living and other ways of being. Yeah, I have thought this a couple times about other questions earlier, and I wanted to say, when I think about people who I think are really good doctors, pretty much every doctor that I meet, I think they're really smart. The people who are really good, I think, and effective are the ones who are good at communicating, and that's quite a different skill set. And at least for me, I spent probably high school, college, and the first half of med school just trying to be smart. And then later, I was like, oh, communication is the thing that is gonna really make me the kind of provider that I wanna be, and I think that's found in other places other than in your neuroscience classes, which are also interesting, but there's other things to do.
I can take a crack at the three-part question. So like the first two questions, I feel like the way that I think that neuroscience has sort of like impacted how I operate in the world, like this is very personal, but because I have this awareness that the brain is really plastic, and that the things that you do in the world and how you talk to yourself will be reinforced in your brain, you know, and so if you talk to yourself in a negative way, then you will feel negative about yourself. And so like I really believe in training my brain to you know, see the world that I want in the way that I want to see it, and yeah, so I think that sort of answers the first two things. And then the most important moment, I think, was the same. Like, we're just having a teacher who actually, you know, encouraged me to keep going. You know, like neuroscience sounds like this fancy, unapproachable field, but I really was interested, you know, and I thought it was really cool. And to have somebody say, like, you can do it, was everything to me. And then also, just in the same vein as Michelle was talking about, like at the end of my undergrad, I did it was a like primatology trip to Tingill Island. This is a place like University of Washington harvests their monkeys from, and so I got to do a research project there. And then every grad school that I applied to said like that was the coolest thing on my application, and I think it kind of made me stand out. So I was, you know, very privileged and fortunate to be able to do that. I was in Indonesia, and so I don't know if you can do something kind of out there. I think it will also help you out.
Okay, and I think the question that we're going to end on is, how do we keep the momentum the field is experiencing while handling current situations at the NSF/NIH?
Can I give a really probably really unpopular answer to this? It might be a good thing that the NIH is getting a little bit of a slap on the wrist and all of that. I think some of the things that we've talked about in terms of like pay-for-play publications. I think a lot of people, and again, I talk to folks who like they get their grant and the first thing they're doing is like thinking about the next grant, and the science was like already done for the first one anyway. And so it does kind of encourage this we're checking boxes to check boxes thing. I think I would say the same thing about doing neuroscience as an undergrad. You kind of like, oh, I'm going to check these things, I'm going to take these classes. There isn't like the underlying sort of goal of it is kind of becomes just feeding itself. They're probably going to fire me because UW's like, but kind of depending too much on one source of authority, I think is kind of it's a little bit dangerous. I think there are a lot of things about it that are good. Vetting, you want to go through a grant process to make sure that you're not just spending money on something that's kind of completely ridiculous. But I do think you kind of need a little bit of a mix. If you think about the whole Elon Musk neuro whatever thing that he's doing, why is it such a big deal? All the things that he's done have been done via NIH-funded things before, but why didn't any of them kind of progress to the point where you have a slick robot and a bunch of neurosurgeons in Arizona wanting to put these things in? It's because it's hard to use, but there's no money from NIH to like make it usable, right? And so I think in terms of keeping the momentum going, those are the people who are doing the research. They're going to want to keep doing the research. I think trying to find alternate ways to do that isn't necessarily a horrible thing. Obviously, how they're doing it is terrible, but that's kind of not what I'm trying to advocate for. But I think sometimes if you're forced to come up with alternate solutions, it's not in the longer term necessarily a bad thing.
So I think, the reality is it's really extremely serious attack on science. So it is unprecedented. It's very freaking depressing that something like this can happen in this country at this time. But the positive side on me says, like my love for science, nobody can take that away. My love for neuroscience, nobody can take that away. And if it comes over, then there will be a rebound, because I was just in Utah where they're just starting a neuroscience program and they don't have enough professors. So there will be a need for neuroscientists coming up. And then out of a crisis often comes opportunities. Like my good friend Toto Oliveira from Utah, who I visited, he was a famous professor in New York doing like modern genetics, and then he moved to Philippines, where he suddenly had no money, no research opportunities, nothing. And he started to isolate toxins out of cone snails, like these mollusks, and found that these toxins can tell you a lot about the neurosciences, because these toxins are very specific for subunits of ion channels. And so out of this desperation that he comes into a country where he couldn't do science, he transformed neuroscience and made it incredibly insightful, because nobody knew that there are so many different potassium channels, so many different sodium channels. But kind of like out of his crisis, something came out. So I don't want to tell a positive story only. We are in a crisis. And I'm so sorry for students that first went through COVID and now through this. But nobody can take away your passion for neuroscience, and it will come back, and then hopefully we can resurrect what we had. I don't know.
Also, just call your reps. Call them and complain. I went recently. I didn't want to spend too much time talking about this because it's nothing to do with neurology or neuroscience, but I went recently to Capitol Hill to talk about tuberculosis funding, actually, with a totally separate group, obviously. But in general, I think as we were talking about this a little bit, like the responsibility of all of us who are in this room to educate the public, which includes sometimes like trying to take actions about it, educating your representatives. I think if you're going to get a degree, people are going to listen to you a little bit more after you put those letters after your name, and it's nice when you can to sort of throw that weight around towards a cause that you care about. And it helps if you're doing it with people that you like. So that's how I ended up in the TB group. My friends are doing it. And we had like 200 people, and we met with like 95% of the Senate, I think. So it's exciting, and that's also how I met Chloe Darlene, because she works with Partners in Health in their advocacy team.
Yeah, I mean, I echo what Andrew says, that like there is processes that are ridiculous at NIH. It can take a year like to get your award. Then if you're a postdoc, you spend the entire year waiting to hear whether you got funded, and it like can be hugely knock you off track. So like, yeah, of course there's a huge amount of reform that could be done in all these processes, and disruption can be helpful for that. But what I look out and I see is an ideological battle against understanding and knowledge and objective truth. And my thought is that the way to fight back is to learn and to teach and to get out there and change people's minds about what matters. So don't give up.
I never, ever write a grant to get money. That sounds weird, but it's really true. I really use grant writing to write down my ideas. What do I want to do? You know, like what is my question? Can I convey this to a reviewer? And maybe my hypothesis is wrong. What kind of experiments do I have to do? So grants is really a good tool to conceptualize your thinking about the problem you want to solve. And of course, it's extremely depressing if you now basically put everything cool in there and then the money doesn't come. Normally the money comes, but the times will come where the money comes again. But I think the process of grant writing itself, don't let this take away from you. So the same thing, don't let those guys destroy your inner strength of doing science. So that is they are not worth that. You know, you come to this life here to change the world, and don't let them destroy that, because they just don't understand this. They don't have passion. They just have greed or whatever. But so don't let them destroy this. So please, that's I think all we have to say, and get through this. You know, if we have to leave the country, we leave the country. But don't let science get your passion for science destroyed.
And with that, let's give another round of applause to our incredible panelists. Thank you so much for joining us tonight to share your knowledge and time with all of us. We now ask that you please remain seated until the drawing of our last raffle winner, since it could be you. We would like to thank ASUW, the Student Activities Office, UW Facilities, the RSO Resource Center, all of the UW staff who helped prepare this venue today, and all of our sponsors. Additionally, thank you to our excellent planning committee and volunteers. Your tireless efforts have been instrumental in bringing an evening with neuroscience to life. Lastly, a huge thank you to all of you for joining us at EWN. It is absolutely wonderful to see so many enthusiastic faces show up. During your time here, we truly hope you were able to learn and have all of your questions about the field of neuroscience answered. So a round of applause to all of you.
Gray Matters has been successful in part because we strive to improve year after year. We will be sending out a feedback form to the email that you all RSVP'd with later. We would greatly appreciate if you all would take the time to fill that out. I'm once again asking for your support as we close out our event. We are entirely a student-funded club, and supporting us means the world to us so we can put on events like this.
All right, so our final raffle prize goes to Runa Dixit. Please raise your hand if you are here. Yay! Congratulations. Great. Yeah, please stop by the stage before you exit, or right now is
fine too, to collect your prize. Yeah, thank you so much, everyone, truly, for spending your Friday night with us. We really appreciate your passion, enthusiasm, and excitement for neuroscience and just science in general. It may not mean much to you, but your participation to us really means the world, inspires us to keep putting on events like this, and keep advocating for a more diverse and inclusive future for our fields of study. And that is the end of our event, but if you would like to stick around for one more little treat, or come up on stage to ask our panelists any remaining questions once the fun video is over, please feel free. Otherwise, we hope you enjoy your time with us today, and we wish you a lovely rest of your evening, perhaps also with neuroscience, and keep in touch. See you next year. Thank you.
When you try your best but you don't succeed, when you get what you want but not what you need, when you feel so tired but you can't sleep, stuck in reverse, and the tears come streaming.